Okay, now we will learn how the lytic cycles happen. Lytic cycle is the cycle or replication of the viral genome that was done by bacteriophage virus T4. This cycle is a cycle of a viral reproduction, which is, in the end of the cycle, it causes cell lysis. This lysis happens to the cell host where the virus lives in, and it causes the death of the cell host. We know that there are 5 stages of this cycle. The first stage is attachment, that will be followed by penetration, biosynthesis, assembly, and ending with detachment or the cell host lysis.
Hello, ladies and gentlemen. My name is Nellis, Nadinda, Putri, Renata, and I will explain the first step, which is the attachment. The T4 page has a complex structure with several tail fibers. These fibers help in attachment of virus to the cell wall of E. coli at the complementary receptor cell. When attached, weak chemical bonds are formed between the receptor site and the attachment, which help the virus adhere to the host cell.
Next, we discuss about the penetrations. Once the T4 patch is attached to the bacterial cells, it is injected its double-strand DNA or single-strand RNA depending on the type of patch into the E. coli cells. The T4 patch releases enzymes that weaken the cell wall of the bacteriums. This helps in injection of the genetic materials of the virus by pressing its sheet against the cells. The empty capsule or virus body remains in the bacterial cells. Some types of patch enter the host cells intact and dissolve their capsule inside the host. This process is known as uncoating.
The third stage is biosynthesis. In this stage, the host protein synthesis stops when the host DNA is degraded by infecting virus. The viral DNA begins to interfere with the transcription and translation of host DNA. The T4 uses the host nucleotides to replicate its own DNA. It is also used the host ribosomes, enzymes, and amino acids to synthesize its own enzymes and proteins. During the biosynthesis, there are no complete phage inside the cell host. Thus, this stage is known as eclipse period.
There are many viral components made in the host cell. These are then assembled into complete viruses. The protein code for phage DNA in case of T4 phage act as enzymes, which are used for construction of new phages. The host metabolism is used for assembly of the phages, which results in the bacteria cell being filled with new viruses. Slowly, the capsids are assembled and the DNA is packaged within the head. Finally, the tail fibers are attached to the complex viral structure.
The last step is release. This is when the viruses are released from the host cell after the assembly of the new viral particle. The phage will produce a certain enzymes that breaks down the cell wall, allowing a large amount of fluid coming in, causing the cell lysis. With the cell lysis, the cycle is complete and the virus will infect another host cell and continuing the process. So that is 5 steps of lytic cycle. We hope you enjoyed this video and see you next time.